Okay. So before the interval, we were in the cell-mediated immunity. And the cell-mediated, in case of cell-mediated immunity, the cells like CD8 T cells or NK cells actually take parts, especially the CD8 cell. And this cell-mediated immune system will work in case of intracellular. Either it is virus, either it is the tumor cell, or either the graft cell, or any kind of facultative cells. And or in case of facultative intracellular, there will be a granuloma formation. So we need to know that what are the facultative intracellular cell, and the antigens are need to be presented by the um, cell to the CD8 T cell. CD8 T cell will bind to the MHC1, and CD4 will bind to the MHC2. And this actually CD8 T cell will not only kill the virus inside the cell, it will kill as a whole the cell. So how many of you actually have the jaundice anywhere in your life? Do you have any jaundice in um, so anyway, so actually in case of jaundice, what happens? The virus infect our liver cell. Virus infects our liver cell, and the CD8 T cell will kill the virus infected hepatocyte. And we know that hepatocyte contains bilirubin, and bilirubin will come into the circulation, and ultimately bilirubin will be raised and jaundice will form. So our CD8 T cell or CD8 T cell will actually kill the virus infected cell as a whole. So that is actually the suicide, assisted suicide. This is called actually assisted suicide. Whenever virus infected cell present with the cell, and this actually assists the suicide of the cell. So CD8 T cell will kill the virus infected cell as well. So this is the one kind of pathogenesis. And like the B cell, like the B cell receptor, or which is the antibody, T cell receptor is also varies. T cell receptor actually also varies to match with various type of antigen. T cell receptor also differs. Also, there are a lot of T cell receptor, a lot of various types of T cell receptor. And if I ask you why is the various type of T cell present, the answer will be genetic recombination. Genetic recombination. We will learn what is genetic recombination in later on. And another type of T cell, apart from CD8 T cell, is the helper T cell. Actually, helper T cell helps. This then by the name you can easily understand helper T cell actually helps. Whether the immune response will go to the cytotoxic T cell, then it will help the cytotoxic T cell, and it will also help the macrophage to enhance the phagocytosis, to engulf the antigen more by releasing the gamma interferon. So helper T cell actually helps the other cells. And in case of B cell, they helps by secreting interleukin 4-5 to produce antibodies. So the function of the helper T cell, function of the CD8 T cell, function of macrophage, everything you need to know, and you have to correlate the things. And that's why the helper T cell actually is the main function, main immune system function occurs by the helper T cell. They are actually the quarterback, or the main cell. Another T cell, another type of T cell, that is the regulatory T cell. Whenever antigen enters into our body, the immune response occurs, but this need to be stopped because there are a lot of cytokines are generated, a lot of tissue destruction occurs by macrophage or neutrophil due to their lytic enzyme. So the immune response must be stopped at some point. And what is the cell that causes the stopping of this immune response? Any idea? Which cell stops the immune response?
T suppressor or regulator T cells.
Like, right, regulatory T cell. So you have to know some of the function of the regulatory T cell. Regulatory T cell is one kind of CD4 cell. It has CD4 and CD25. Some of the features actually you need to know. CD20, they have the unique marker CD25. They have a unique marker of interleukin high affinity. They have the high affinity interleukin 2. Do you know what is the function of this interleukin 2?
Activation of T helper cell.
activation of T cell. And the regulatory T cell have the high affinity of interleukin 2. That means they will take the all of the interleukin 2 surrounding all of the interleukin 2. They have the higher affinity of interleukin 2 receptor in comparison to helper T cell. So they will suppress the other cell, other T cell by high affinity interleukin 2. They will absorb all of the interleukin 2 by themselves. This is how they regulate the immune response. And they also release some of the cytokine, immune inhibitory cytokine. If I ask you what are the immune inhibitory cytokine, can you name it? What are the immune inhibitory cytokines released by regulatory T cell? TGF beta, tumor growth factor beta, and another one is interleukin 10. Can you see what I am writing?
Yes, sir.
Okay, so the cytokines also, some of the cytokines, they are immune pro-inflammatory cytokine. What are the pro-inflammatory cytokine? Again, actually, these are the frequently asked question in the residency exam. That's why you need to know what are the cytokines that are takes part in the inflammation. What are the cytokine that are inhibitory? What are the pro-inflammatory cytokine? If I ask you what are the pro-inflammatory cytokine, or in case of inflammation, what are the cytokine? Interleukin 1. Then what I have right? Interleukin 6 and TNF alpha. Interleukin 1, 6, TNF.
Yes,
These are the pro-inflammatories. Any kind of inflammation, these cytokines will be raised. Any kind of immune inhibitory, these are the immune inhibitory cytokine. Okay. And we need to know how the antigen is present, how antigen is present. Like in case of virus, can you see the screen or not?
Yes, sir.
yes, sir.
Wait, I'm not seeing any screen. Achha, why I cannot share? One minute. What is in your screen now? So whenever CD8 T cell or CD4 T cell actually um, uh, attaches an antigen, then the antigen must be need to be processed and present to the antigen. And this presentation is done by antigen presenting cell. Some of the antigen presenting cell, they are professional. We call it as a professional antigen presenting cell. Some of the antigen presenting cell, they are non-professional. We all know the example of professional antigen presenting cell. What are the example of non-professional antigen presenting cell? Any idea what are the non-professional antigen presenting cell? One or two example. Can you give one or two example of non-professional antigen presenting cell? Okay, why we call it professional antigen presenting cell? Why we call these cells are professional antigen presenting cell? Because the antigen, whenever enters into the body, like in macrophage, they will engulf, they will process, then present. This processing is done in case of professional antigen presenting cell. And they will secrete some of the cytokine that will enhance the immune system. That is another role of the professional antigen presenting cell. So the processing, and they will present the antigen by the help of MHC, MHC2. That is professional antigen presenting cell have the MHC2 for the presentation. So that are the three reason they are called that professional antigen presenting cell. So this presentation, this MHC presentation is actually a billboard, whether to kill whenever antigen is presented in virus, like virus infected cell presented with the MHC, they will be act like a billboard that just kill me, kill me, and the CD4 will T cell will come and kill the cell. So this presentation is actually a billboard that whether the cell wants to kill himself by the cytotoxic T cell, or whether the cell wants to help, the antigen presenting cell wants help, that is by the helper T cell, either to produce the antibody or to go through the go to the cell mediated immunity. That is the antigen presentation. So the activation of adaptive immune system, we all know that innate immune system and adaptive immune system are interrelated. Like macrophage, can you see the picture? What does the picture shows? The macrophage or the log box? These are the picture. What are you seeing? What are the slide actually you are seeing right now? What are the headline? What is the headline of this slide?
Antigen presenting cells is presenting antigen to the helper T cell.
Activation of adaptive immune system. This is the headline you are seeing, na? Activation of adaptive immune system. Okay. So now the adaptive immune system actually um takes part or activated, and we know that the innate and adaptive immune system are interrelated. Like phagocytosis is a part of innate immune system, works by macrophage, and after antigen phagocytose, they will be processed, they will be fragmented, they will be processed and presented to the helper T cell by the MHC, what? MHC2, MHC2 to the helper T cell, by the MHC2 to the helper T cell. This response is the first. This response is we call it as a first signal. This response is, we call it as a first signal. There is another signal is present, and you need to know what is the signal. This is the antigen presenting cell. It has the B7. And this is the CD4 T cell. It has the CD28. This is the signal. We call it as the second signal. Both are the signals are needed for the proper immune cell activation. So why two signals are needed? Because immune system activation in immune system has a lot of deleterious consequences. It will destroy the cell. It will secrete a lot of cytokines, which will damage, or vascular endothelial damage can occur. So it need to be checked, and this is the double checking system. So the second signal and first signal, both of the signals are needed for the proper immune activation. And there are some drugs actually developed to block this second signal to get our appropriate immune suppressive condition. So in cancer drugs, there are a lot of drugs actually developed to block this second signal. And whenever the second signal is blocked, what is the condition of the cell? What is the condition of the situation? Anyone know it? Energy. We call it as energy. Tolerance. In case of tolerance, you will heard the term of energy. You will heard the term of energy. So what is energy and how this energy occurs? You need to know. The energy is actually blocking whenever the second signal, that is the B7 and CD28, do not work, or that is the energy. That is the energy, and ultimately further immune response will not occur. This is the energy. Some of the things actually you need to know. There's a clonal expansion or clonal multiplication. Suppose this is the cell, this is the B cell, and this is we know that the B cell have a lot of type of immunoglobulin present in their surface. So the antigen, whenever antigen comes, it will try to match the cells. And suppose this is the cell, actually B cell, this is the B cell where the antigen matched, and this B cell will multiply and this clone will be multiply, and so this is the clonal expansion or clonal selection. Clonal selection is when the particular antigen will bind to the B cell, that is the clonal selection, and that type of B cell will multiply, that is the clonal expansion, and will produce an plasma cell and antibody, and that antibody will ultimately bind to the particular antigen. So this is the clonal selection and clonal expansion. um, uh, I think you got it, or any kind of problem. Do you understand it? What is clonal selection or what is clonal expansion? One type of clone will be got selected, antigen will fit that type of cell, and that cell will be clonally activated. A lot of clones or a lot of that type of cell will multiply. That is the clonal activation or clonal selection and expansion. Do you understand? Anyway, so innate and adaptive immune system difference.
sir.
This is the written exam. This is the written question, that what is the innate immune system and what are the adaptive immune system, the difference between the innate and adaptive immune system. And there is some what is the PAMP, what is the PRR? Any idea about this? What is the PAMP, what is the PRR? We will learn about the what is the PAMP and what is the PRR. Suppose we are calling that innate immune system are non-specific, but this is not actually true. Innate immune system, we can say it as a less specific. That means innate immune system, sometimes innate immune system can mm, uh, match or can match a particular pattern. Suppose this is a cell. This is actually a innate immune system cell, and there is a receptor. We call it as a, like, this is a toll like, TLR means toll like receptor. Toll like receptor. It will bind some of the PAMP. PAMP means pathogen associated molecular pattern. Pathogen associated molecular pattern, like flagella. It can be flagella. It can be lipopolysaccharide, and that flagella will bind to the TLR TLR5. Lipopolysaccharide will bind to the TLR4. So this TLR is the pattern recognition receptor. We say that TLR is a pattern recognition receptor, and this flagella or lipopolysaccharide, these are actually the PAMP, or damage associated molecular pattern, DAMP, whatever you say. So you can say that these are the innate immune cells, but they can actually not working as a non-specifically. Sometimes they are less specific. They can actually bind to the flagella, or they can bind to the lipopolysaccharide. So we can say it is a less specific, but it's okay if you write the as a characteristics, innate immune system are the non-specific, but actual term will less specific. And we know that what is PAM, what is PRR, what is DAMP. These are the abbreviation we need to know, right? So innate immune system actually, whenever we enter into the forest, then tiger comes, and our response will be to run, and that is the innate immune system. As a whole, we will run it. Second time, if we go the same forest, we'll now be prepared and act specifically to kill, try to kill the tiger or something. So that is the specific response. So innate immune system and adaptive immune system, actually the main purpose is it is actually working as a holistic method. Right? So characteristics of innate immune system, you need to, these are the MCQ questions. আচ্ছা আমরা একটু দেখি যে এটা পারি কিনা। Characteristics of innate immunity, it is a rapid onset of action. Is it true or false?
True.
Rapid, true, right? Is that whenever antigen enters into our body, our neutrophil or cells will try to get rid of even the skin pH of the gastric. They are also part of innate immune system. So this is a rapid onset. Long-lasting immunity, true or false?
False.
False. Good. First and second body defense. True. We can say true. First line, second line body defense. We can say true. Acute phase protein play important role. Is it true or false? What is acute phase protein? What are the acute phase protein? Can you give an example?
C-reactive protein,
C-reactive
alpha-1
protein.
antitrypsin.
Fibrinogen. The acute. What is the function of acute phase protein? they have a lot of function, but you can say that these are they are kind of help the cell to phagocytose. They are kind of opsonizing to clear the antigen from the body. So one of the function is as a opsonizing particle. Acute protein, the CRP, they are actually part of innate immune system. Immune response is specific. True or false?
False.
False. So immunity we can divide humoral or cell mediated by the action. By action, acquired immunity is divided humoral and cell mediated. Humoral and cell mediated immunity, these are divided due to action. This is the action, and in case of um, um, uh, requirement, or in case of acquired immunity by acquired, we can divide it by natural and artificial. We all know what is the example, so I'm not going details. What is artificial, what is innate, and what is active immunity, what is passive immunity. Example of what is the example of passive immunity? Passive immunity, artificial passive immunity. What is the example of artificial passive immunity?
TIG, tetanus
TIG.
immunoglobulin.
Good. TIG, that means immunoglobulin. What is the benefit of this passive immunity? Artificial passive immunity. Benefit. Suppose a patient comes to
Immediate.
you. Suppose the patient comes to you with a rabies dog bite. You will give the patient immunoglobulin, rabies immunoglobulin plus rabies vaccine. Rabies immunoglobulin is a passive, artificial passive. So what is the benefit of this artificial passive immunity or immunoglobulin?
Rapid response against inoculated virus.
Rapid, it will clear up the it will clear up rapidly the virus. So the action is fast. What is the disadvantage of the passive immunity? It is short lasting. So what is the and you will also give the vaccine. So what is the benefit of the vaccine? Giving the vaccine, what is the benefit of the vaccine?
Long lasting immunity.
Long lasting. What is the disadvantage of the vaccine?
It takes time to develop
Good.
antibodies.
It takes time. Some of the case, there is a active passive, both immunity are produced. What are the case? Can you both active passive? That means both active passive immunity we give. Rabies in an example. What are the other example? Tetanus. Good.
Tetanus.
Good. Are
Tetanus.
tetanus. Good. Diphtheria is another example.
Diphtheria.
Botulinum, sometimes botulinum is another example. Hepatitis B, hepatitis A, these are the example where both active passive immunity are used. আচ্ছা, আবার চলে গেল। Again, running out of time. So I will not continue much. Just so this is these are the things actually. Whenever antigen enters into our body, it is engulfed by the macrophage and present with the helper T cell. Helper T cell will either go to the cytotoxic T cell, or helper T cell will divide in the B cell, depending upon the intracellular or extracellular pathogen. If it is extracellular, then the humoral response will happen. If it is intracellular, then the cytotoxic T cell will kill the cell. And both the cases, memory cell will generate it, and the memory cell will produce the secondary immune response. So do you have any question regarding this overview? Actually, immunology, I cannot finish it within 1 or 2 classes, but I hope that So let's stop it. Okay, I have 8 minutes. Then the class will be over. Let's. So the cells of innate immune system, cells and organs of innate immune system. What are the things actually we need to know? Can you see the slide is changing? Cells.
Yes, sir.
Okay. So in case of cells, actually, we need to know every function of the cells. What are the cells? What are the function of the cells? What are the innate immune system cell? What are the adaptive immune system cell? So you need to know the what are the function, every function, and sometimes we need to know the characteristics of the cells too. It's like in case of macrophage, we need to know everything about the macrophage. What are the receptor of the macrophage? What are the receptor of the, and sometimes what are the receptor of the neutrophil? What are the receptor of the T cell? What are the receptor of the B cell? So, and what are the function of the each receptor? So that are the cells and organs we need to know. Some if we see the primary lymphoid organ, secondary lymphoid organ. So in case of primary lymphoid organ, the T cell and B cell, and the two of the important function of primary lymphoid organ is um, production and the education. Production of the T cell or B cell, and they will not only the produce, but also they will educate the cell. So my question is how they educate the cell, educate the whether it is T cell or B cell. Anyone can give the answer how the education occurs?
Positive selection and negative selection.
Okay, fine.
Positive and negative selection.
Before the positive and negative selection, this is the thymic. This is the thymic medullary epithelial cell, which is a non-professional antigen presenting cell. It will present the antigen to the T cell into the thymus. This is happening into the thymus. This is happening in the thymus. And now our T cell will just bind in a low affinity to the antigen. This is the antigen actually with either heart or lungs or kidney. That means every cell, every cell of our body, every cellular protein of our body will be presented by the medullary epithelial cell to the T cell. Now the T cell will react either with low affinity. If it is low affinity, then what will happen? This test cell will survive. This is the positive selection. If it just bind with low affinity, then the cell will survive. It is bind if it is the T cell binds with the high affinity, then what will happen? Can you see the
Negative
whiteboard?
selection.
Negative. Can you see the whiteboard? What I have written? Can you see the whiteboard?
Yes, sir.
Okay. So if the T cell will bind by the T cell receptor to the medullary epithelial cell with the very high affinity, then the T cell will be apoptosized or T cell will be deleted. This is the negative selection. This is the negative. My question is how various types of cells are presented by the medullary epithelial cell to our T cell into the thymic education. We all know what is positive selection. We all know what is negative selection. But my question is how various type of cells are presented by the medullary epithelial cell to the T cell during thymic education during the very early stage of our life. Do you know the answer?
Genetic transcription of various proteins.
Genetic transcription of various protein. You are close. You are close. Actually, the answer will be AIRE. Do you know what is AIRE? Autoimmune regulator. Do you know what is AIRE? AIRE. This will be I have this is actually told you in the tolerance class because I did not take the tolerance class. Okay. Okay, just take the question into your mind because it will take time to elaborate the thing that actually every organ protein will be presented during the very early stage of our life to the T cell, so that the T cell will recognize, so that T cell will know the cell, this is our cell. And then the fate of the T cell will be determined whether the T cell will be destroyed by negative selection or whether the T cell will be survived by the positive selection. Most of the cells will be destroyed, 95%, and only 5% will survive, which is the positive selection. Okay, AR is the autoimmune regulator. This is the transcription factor. Actually, this will present. This will generate our DNA to the. This will generate. This is the transcription factor actually responsible for presenting various types of our body cell to the T cell. This is the short answer. And whenever, so what happens is the AIR fails to do its function. What will happen? Now AIR
Autoimmune disease.
good. Auto now AIR will fail because now the heart cell, lung cell, kidney cell cannot be identified by the T cell. So the T cell will survive and go to the periphery and will destroy the heart, lung, kidney, and that will cause the autoimmunity. Autoimmune, that is called the autoimmune polyendocrinopathy. Autoimmune polyendocrinopathy or autoimmunity, one type of autoimmunity. So if AIR fails, then the autoimmunity develops. So all of these things, thymic education is actually during the very early stage of our life. Acha. Acha, I have less than 1 minute. So I think do you want to continue, or I'll finish the lecture? It will be very monotonous for you to continue the lecture actually for a long time, because there are a lot of topics actually. But actually what I want to tell you that whenever we will study, actually we will go as a whole picture. That is the main thing. If you want to understand the immunology well, that is the one thing you need to keep in your mind. And definitely the picture visualization, that is important. And let's see how I can provide you something like lecture note. I'm working on it. Hope that will take some time to provide. So now I'm going to finish now. I have less than 1 minute, so maybe another time we will meet. Okay, do you have any question?